Good afternoon. My name is Jack Burns. Thank you for coming to my senior project presentation. For my senior project, I designed and 3D printed biology models. My essential question was, what are the steps needed to design interactive models of 3 biological processes to facilitate the education of AP Biology students at Millis High School? That's a long way of saying I was just going to design models of DNA replication, transcription, and translation. Anybody that's taken biology freshman year or AP Bio has probably heard of those processes or is familiar with them. Just a quick outline of my presentation. I'm going to talk about why I chose this topic. Then I'm going to go into the 2 mentors, talk a little bit about them that I had for this project, followed by my timeline from start to the year to now, which will lead into my final product. Then I'll discuss some successes of this project and then some difficulties and some challenges I ran into, and then what I would do next time. I chose this project because I took AP Biology my junior year, and I learned about these 3 processes. And there's a lot going on in these processes. There are enzymes, which are the individual pieces that allow for this entire thing to work. So it's a very difficult thing to get a grasp on, and it's easier when you have a visual demonstration. Mr. Alcanada used to have Adobe animations that would show these processes, but Adobe is no longer compatible with current software, so they don't work anymore. I was inspired to help future students have an easier time learning these processes, which is why I took on this project. And I'm also a STEAM scholar, so it had to be STEAM related. I was prepared to take on this project because I had a deep understanding of DNA replication, transcription, and translation, because I took AP Bio. And then I also took a 3D computer-aided design virtual high school class over the summer, actually to fulfill a STEAM scholar requirement. And it taught me how to use Autodesk Inventor, which is the computer-aided design program I use. This is actually a project I worked on over the summer. I also had a really good relationship with Mr. Alcanada, so I knew it would be very easy for this project to work between the 2 of us. I have 2 mentors, Cassandra, or Cassie Probert. She is my cousin. That's actually a picture of us at our Christmas party. And she just graduated from UMass Amherst with a degree in biology. And her kind of contribution to this project was explaining the key takeaways to these processes as a whole, but also what each individual enzyme is responsible for doing. So I tried to design each piece to reflect those big ideas. My second mentor was Michael, or Mike Gilnet, and he graduated from Wentworth Institute of Technology with a degree in engineering. And he helped me to kind of craft a realistic timeline of this project with his background in 3D printing and that design process. I knew it was going to be a time-consuming project. However, I did not know just how time-consuming it would be, which is why it was helpful to kind of craft that timeline. Now, going into that timeline, I started by having the idea approved by Mr. Alcanada and also Ms. Cheney. And then I reached out to Mr. D, who is the computer teacher at the school. He's the one who runs the 3D printers, because obviously I would need those to 3D print. Then September to December, the first part of the year, I brainstormed and finalized my ideas for each piece. I would have different ideas and I would combine some, I'd have to scrap some, and end up coming up with one idea that I really, really liked. I also brainstormed dimensions for each model, which consisted of me just like holding a ruler in my hand and trying to visualize how big I wanted each piece to be. And then I had those final sketches reviewed and approved by Mr. A. So these are the designs that I have in my notebook. This is the first one. I just wanted an easy way to store all the pieces together, but I also wanted that there could be a base for all of these pieces to rest, because obviously there needs to be something to hold it up in order for this process to work. So here's my first design for the box. The first process is DNA replication. I have my 2 pages of notes there, along with, off to the side, these are pictures from the Amoeba Sisters videos. Those are videos we were watching this class. So it just shows you there's a lot going on with the enzymes and as well with the process. Transcription is the second process. However, the way it functions is very similar to DNA replication. So I was able to use a lot of the same pieces and the same concepts and apply it here, which is why my notes are more just writing and not designs. And the third process was translation. This one is where what's created from transcription is a messenger RNA, and that messenger RNA is then sent to the factory of the cell, the ribosome, and that messenger RNA is read by that ribosome, which then creates proteins in our bodies. Then the second part of the year, January to March, was all about designing those pieces on that computer-aided design program, and then also just modifying to accommodate any new ideas I had or any limited abilities that I might have since I'm not a professional designing these pieces. Here are a few of them up here. This piece over here is the DNA strand, so this is actually a bunch of those tied together, which makes it very easy for it to bend and pull apart, which you'll see is important when I get to the next part of my presentation. Then down here are 3 enzymes that are important in DNA replication: helicase, DNA polymerase, and ligase. All of these have their own specific functions. You can see this is what they look like online, this is what they look like when they are printed out, which is pretty cool. Then I have, these are what create the chains for the protein, the amino acid. And then this is the ribosome up here, which is this big piece right here. So when it comes to 3D printing, it ranges a lot in how much time it takes. Pieces like this could take about a half hour. This one took 36 hours. So it is very time consuming in just trying to get things printed. March to April is when I printed out prototypes of each piece, making sure everything worked together. And actually during this time is when the Millis 3D printers broke down and they were offline for about a month. So I had to outsource a lot of my printing to Wellesley High School, where my dad works, and then also Medfield Public Library later on. Then once I got those pieces back, I edited them a little bit, redesigned if needed, and then would just keep doing the same thing, printing, see what works, see what needed to be changed. Then April to May, right about now, is when I finished printing all of the pieces. I painted the individual pieces that needed it, because with 3D printing, you can only print in one color of plastic filament. So when it came to these DNA strands that are very colorful, all of that was like 2 layers of white paint and then 2 layers of the color it needed to be. Then I magnetized all the necessary pieces. These magnets I attached with super glue, which was a bit of a challenge, which I will discuss later. I still have super glue on my hands from trying to glue these very small magnets onto very small plastic pieces. And then that leads me to my final product. I do not have it here with me today because the glue is still setting, but this is kind of an idea of what DNA replication would look like. I have the helicase, which unzips the DNA strand, and you can kind of see how, with the structure, it helps you pull apart those 2 strands. So my successes from this project, what I think went really well, was I was able to refine my computer-aided design skills that I learned over the summer. And then I also learned how to 3D print, which was really interesting. And I think that's something I'll try to continue doing more with. I also got these functioning models that Mr. A actually really likes. So after everything is set, the glue is dry, and I know nothing will fall apart when I try to transport it, I will bring those to school and Mr. A will have them for next school year. Also along the way I came across many obstacles, but I was able to quickly overcome those and continue on. Luckily that did not cause too much of a hindrance to the timeline I had set up. A few challenges from this project. Autodesk Inventor is not used as much in the business world as I had thought, meaning my mentor, the people he worked with, even engineers that he knew through other jobs, none of them had ever heard of the program. So for a second, I thought I'd have to learn a new program. I tried, didn't work out very well, but at the end of the day, I was the one designing the pieces, so I was able to just go back to this program. The broken 3D printers was obviously a challenge. having to switch to other printers and rely on those meant that a lot of my prototype pieces that I printed out had to be done in bulk. So I couldn't just print out one and see how it worked. I have to print out like the 10 I thought I would need, realize that the sizing wasn't right, have to edit it, and then send it back to be printed, because I was just trying to get those pieces as soon as I could. And then magnetizing individual pieces, as I'd already talked about a little bit, was difficult. As you can see in this picture right here, these two magnets are stuck together, but not in a good way. Those two magnets are super glued together. So I have to find a way to disconnect those two magnets and replace it with another one, so there's not double magnets on the strand, so it works the right way. I think in the future I would rely on Medfield Public Library to 3D print, just because the ones in Millis had some difficulties. Once it was fixed, it broke down again. I would also break up the pieces and design them a little bit so there was less painting and I could print them out in one color and just glue them together after. And I would also try to go model by model when printing, so I wouldn't have like all of these files that I would send in and then not know which one goes where. I have quite a few thank yous. First to my parents, because they helped me with buying materials, finding new places to print, painting, adding the magnets, literally every step of the way they were there to help me. Also thank you to my two mentors, Mike Gilmet and Cassie Probert. Thank you to Mr. A and Mr. D for helping me out with this project in school. And then also thank you to Ms. Copese, Ms. Keeney, and Ms. Boise. They acted as my in-school mentors if I ever had a question that I needed to quickly answer. And then also thank you to the Wellesley High librarians who allowed me to use their 3D printers when the ones in Millis were not working. Here's my works cited, and that's the end.